Hello everyone, my name is Jessica Chalk and today I will be talking about the gene DNAJC7 and how it may be implicated in ALS. So a quick overview of what I'll be talking about today. First, I'm going to give a brief background introduction to the central nervous system and what can go wrong in the central nervous system that leads to ALS, and talk about some of the genetic risk factors associated with ALS. Then I'm going to hypothesize that DNAJC7 should also be considered a genetic risk factor in ALS. I'm going to show some clinical studies that show that DNAJC7 is a possible pathogenic mutation in ALS, and then suggest some future directions to provide further evidence of this relationship. So the central nervous system consists of neurons, the brain, and the spinal cord, and its role is to process information from the environment and respond in the appropriate way. The central nervous system controls sensory integration, information processing, and motor output. When one of these roles in the central nervous system is compromised, it is considered a neurodegenerative disease. Neurodegenerative diseases can affect one or all aspects of the central nervous system, and causation for neurodegenerative diseases is largely unknown in the scientific community. One neurodegenerative disease that is still being studied is amyotrophic lateral sclerosis, or ALS. ALS is a motor neuron disease that weakens muscles and slowly causes muscle paralysis throughout the body. ALS has roughly a 5 to 10% familial type of ALS, which means it is hereditary, but most cases are sporadic, meaning that they occur through random genetic mutation. ALS occurs when proteins aggregate in the brain and kill off motor neurons, and the toxicity behind the protein aggregation is still unknown, but research has suggested that genetic and protein mutations may be the reason why protein aggregation occurs. In ALS, several genes and proteins have been identified as pathogenic mutations, the main ones being SOD1, FUS, and C9orf72. The SOD1 gene codes for the SOD1 protein, and this protein is an enzyme that is responsible for breaking down toxic charged oxygen molecules that cause DNA damage. So when a mutation of SOD1 gene occurs, the SOD1 protein aggregates and misfolds in the spinal cord. Another gene associated with ALS is FUS. The FUS gene codes for the FUS protein, which is a DNA and RNA binding protein that is involved in transcription regulation, RNA splicing and transport, DNA repair, cell damage response, and the formation of dynamic ribonucleoprotein granules. When mutations in FUS occur, this leads to an accumulation of the FUS protein in the cytoplasm and the formation of stable ribonucleoprotein granules, which can lead to inclusion bodies and neurotoxicity. The last main gene associated with ALS is C9orf72. This gene codes for the C9orf72 protein, which is believed to be responsible for the production of RNA from genes, the production of proteins from RNA, and the transport of RNA within a cell. When C9orf72 mutations occur, nucleocytoplasmic transport is disrupted and stress granule assembly occurs, which leads to protein aggregation. As stress granules are composed of RNA and proteins, stress granule assembly related to C9orf72 is considered a key hallmark in ALS pathology. So one thing these genes all have in common is that research suggests that the mutations involved in ALS work together to produce neurotoxicity. This is why it's difficult to identify singular genetic mutations implicated in ALS. So an approach to finding these implicated genes is to look for gene variants in ALS patients and identify the gene's function. If a variant causes a disruption in the gene's function, then it's worth further looking into, which is why today's presentation is on the gene DNAJC7. DNAJC7 is a gene that acts as a co-chaperone for heat shock proteins 40 and 70. One role of heat shock proteins are that they regulate proteins and control their aggregation. So another hypothesis behind ALS pathology is that motor neurons have an impaired ability to call for a heat shock protein response. The role of DNAJC7 in humans has also been previously studied, as this gene is important for proper liver function, kidney function, and tumor suppression. As DNAJC7 is important in heat shock protein activity, the relationship between DNAJC7 and ALS is starting to become of interest in the scientific community. So, DNAJC7 being related to ALS was first discovered through the world's largest exome case control study to date. In the study, DNAJC7 in ALS patients had exceeded genome-wide significance because it had multiple distinct protein truncating variants, a variant that causes disruption of transcription and the shortening of proteins, which leads to abnormal protein activity. Additionally, through immunoblotting, the authors discovered a depletion of DNAJC7 in fibroblasts in an ALS patient who carried the P.R156 TER protein truncating variant. As DNAJC7 is an important co-chaperone for heat shock protein 40 and 70, variants in this gene can lead to improper protein maintenance, such as mistakes in protein degradation and the synthesis of new proteins. After DNAJC7 was discovered as a novel ALS gene in this paper, clinical studies were conducted to provide further understanding of its role in ALS pathology. All clinical studies to date connecting DNAJC7 to ALS have been conducted in Asian countries, and each study specifies that their findings may only apply to Asian patients and should be verified in European and Caucasian patients. The first clinical study to come after the genome paper verified DNAJC7 as a mutant gene in ALS patients with rapid progressive ALS. Out of 325 ALS patients, the authors found one patient who had a heterozygous DNAJC7 frameshift mutation, P.Q134RFS*6. This specific case was SALS, however, so the authors could not do further research on the heritability of this mutation. But it was concluded that DNAJC7 should be considered a risk gene in ALS. The second clinical study discovered two rare heterozygous missense variants in DNAJC7, P.K137R and P.N369T. But they were not considered pathogenic in Chinese ALS patients. While the study suggests that DNAJC7 is not pathogenic in Chinese ALS patients, they should still look at genetic variations in Caucasians and Europeans, as it may still have clinical applications and should still be further researched. The last clinical study to date identified a rare pathogenic variant in the DNAJC7 mutation, P.R238G. Right here. And this is also a SALS patient. This patient exhibited rapid progression of the disease at a relatively younger age, around 43. So the authors postulate that DNAJC7 mutations may occur in younger onset ALS patients, a smaller percentage of ALS patients. Again, the authors of this study suggest that further research should be done with a larger ethnic sample so that more patients can be verified. So to conclude, I believe that there is some evidence that mutations in DNAJC7 may play a pathogenic role in ALS. While the clinical studies I discussed had varying results pertaining to their DNAJC7 ALS patients, I believe that this may be another example of ALS-related genes working together to produce neurotoxicity. And results varied in these clinical studies because other ALS patients had other genetic mutations. Improper functioning of heat shock proteins in DNAJ genes can lead to hallmark protein aggregation seen in ALS and other proteinopathies. So I would like to suggest some future directions to provide further evidence of this relationship. First, I believe that more ethnically diverse samples should be screened for DNAJC7 so that the functionality of this mutated gene can be further understood. Secondly, I do suggest taking a step back and conducting experiments in human motor neurons and other model organisms to isolate the function of DNAJC7 in ALS. One model organism of choice is Drosophila. Drosophila models have become a useful tool in studying neurodegenerative diseases and genetics because they share roughly 75% of their genes with humans, so they have many gene homologs that can be manipulated and studied to determine their functionality. Scientists are able to verify genome sequences in Drosophila from specific phenotypes. As technology in this field has progressed, and the verification of gene sequence can be done by inserting genes onto specific chromosomes that phenotypically express a desired characteristic, for instance, in the eyes or in their flight patterns. This technology has existed for a few decades now, but recent strides in transposon technology have made it easier for scientists to insert a gene sequence into Drosophila and maintain the gene sequence in future generations. This technology is called Meidos-mediated integration cassette, or MIMIC, and it works by carrying a dominant marker in a gene trap cassette flanked by two inverted Phi-C31 ATTP sites. This technology allows for almost limitless gene engineering and gene modifying applications, and this should be considered as a future direction for ALS-related DNAJC7 research. Lastly, while this was just a literature review, I still wanted to thank my lab at Mayo Clinic, these people here. It's because they gave me the idea to look into DNAJC7 in ALS, because we may actually possibly work on this in our lab. And of course, I have to thank Dr. Ockrider for being my mentor and helping me write my paper and help me along the way to make sure that this is the best presentation I could possibly give. Thank you so much for listening to my presentation.